Alright, so we'll start today. What we are going to start with is this topic of random walks in biology, and so I'll start by describing sort of a model system, the canonical system for random walks, which is E. coli diffusion. But before I get into that, I would encourage all of you to have a look at this book, Random Walks in Biology, by Howard Berg. He is one of the proponents of this field beginning in the 1970s and so on, doing a lot of work on analyzing bacterial motility, how do bacteria move in different environments, trying to tease out the stochastic variables and the relevant quantities through experiments and theory. And this book is a very nice sort of introduction to all of that. It's fairly simple, in fact. It contains some experiments, some theory, but most of it is, at least to this audience, should be fairly easy to read and understand. So I encourage all of you to sort of have a look at this book. So let's get into it. So here is this E. coli bacteria. So I thought I'll just introduce this system, because often, as you'll see as we go along, often when biologists try to understand phenomenon, they look at some model systems, model organisms in that sense, which various parts of which have been very well characterized, and we try to understand the properties of those systems, because often structure and function is conserved across organisms. So if we understand how certain processes happen in simple organisms, we can take that understanding and try to build together the understanding for more complex organisms. So E. coli is one such model system. It is and of course, all of you have heard of E. coli and so on, but still, so this is a bacteria. Okay? It's a class of organisms called prokaryotes, in that there is no, if you think about the human cell, a human cell or any eukaryotic cell, you have the cell membrane, and inside the cell membrane is your nucleus. So this is your cell, and inside is your nucleus, and your genetic information, your DNA, all this chromatin that we talked about, lies inside the nucleus. So it's separated from the cell, right, from the cytoplasm. So this is my cytoplasm. Inside the nucleus is my nucleoplasm, and these two are separated by this nuclear membrane. So, and then the transcription happens inside the nucleus, the proteins are produced, and then they come out into the cytoplasm and do whatever business that they needs to do. On the other hand, in these sort of eukaryotic organisms, there is no nuclear membrane. The DNA is sort of intermixed with the cytoplasm, and this lighter segments over here is your DNA. The darker segments are the non-genetic component, the cytoplasm. So, and there is no wall, there is no membrane that covers that separates these two compartments. Even then, of course, you will see that there is a fairly effective compartmentalization that arises, in that it's not completely black and white, completely homogeneously mixed together. So there is still some segregation, but that segregation does not happen through a membrane compartment. It arises spontaneously due to other physical forces. Okay? Right. So this is your DNA, and this is E. coli. Just to get an idea about sizes, about the scales that we are talking about, these E. coli bacteria are of the order of microns. So roughly, generally, the length of this E. coli body is around 2 microns, and it's roughly around a micron wide. Of course, there is variability. Whenever we talk about these numbers, nothing is set in stone, but this is roughly the scales that we are talking about, a few microns when we are talking about E. coli. On the surface of this E. coli, you'll see these sort of hair-like structures. These are called pili, and what they do is that they help bacteria adhere to surfaces or other bacteria, for example. So they help in colony formation, biofilm formation, and so on. What is of particular interest is these other sort of long structures that you will see over here. These are the flagella, and that is what we'll talk about mainly today. So these flagella are sort of filamentous polymeric assemblies. So these are your flagella. These are fairly long. These are longer than the cell body, and what these do is that these help the bacteria swim in their natural environment. So they are responsible for the locomotion of the bacteria. Before I move to the flagella, just to give you an idea, this is helpful in various contexts. We see sort of these pictures of cells and so on. but it's good to realize that the inside of the cell is an extremely crowded object, is an extremely crowded environment. So this is a cartoon in some sense, but what this cartoon aims to show you is that there is not a lot of free space inside the cells. So when we say that things move inside cells, when we say that proteins are transported from somewhere they're produced to somewhere where they're used, they're not moving, they're just not moving through air, or they're not just moving through a fluid medium. They're moving in a complex, crowded environment which is filled with other proteins, which is filled with these polymeric filaments, microtubules, actins, and so on and so forth, and they have to navigate their way through this complex, crowded environment. This book, this Physical Biology of the Cell, this Rob Phillips and company book, it does a very nice sort of number estimation, just to give you a sense of the numbers that are involved. So, this is, like we said, so this cell is of the order of microns, so the volume is roughly around a few micron cubed, right? And then if you look at the number of particles of each species that is present, so for example, DNA is of course 10 to the power of 6 base pairs. If you think about proteins, there are again 10 to the power of 6 proteins. Okay. Ribosomes, which are these particular type of proteins that help in this transcription and translation process, these are 10 to the power of 4. RNAs, some thousands of RNAs, of course, billions of water molecules. Proteins which are embedded on this membrane, that's also about 10 to the power of 6. Many inorganic ions, many lipids which are again embedded on these membranes, again 10 to the power of 7. So these are very, very large numbers, okay? And all of these billions and billions of ions and small molecules and large molecules and so on are contained within this micron cubed volume. Okay? So the density of these cells are extremely high. And whatever physical processes and chemical processes happen inside these living cells must happen in the background of this extremely crowded environment. Whenever I talk of these numbers are not set in stone. So, for example, here is some census of the number of proteins. I think I forget whether it's different cells or cells at different points of time in their cell cycle, but regardless, what you'll find is there is a huge variation. Not just a twofold or a threefold variation, but an order of magnitude variation in these protein populations if you look at different cells or at different points of time. Okay. So these are of course fluctuating quantities, but still just to give you a mean estimate of how crowded these cells are, it's good to keep in mind these sort of numbers. All right. So what I'll focus on is this flagellar structure over here, which is, like I said, responsible for E. coli locomotion. So here is an E. coli. Again, this is from Howard Berg's work. Like I said, a lot of the early work on bacteria. Berg is one of the principal people. So what happens is that on an E. coli cell, you'll have multiple flagella. All right. So you'll have multiple of these. Flagella. And these flagella, these are protein complexes. So these are proteins, these are made of many, many proteins. I have a video later on which I'll show. But these flagella have this molecular motor that is embedded inside this membrane. So here is a molecular motor, and the flagella is attached to this molecular motor. And what this molecular motor can do, so here is my molecular motor, this bacterial flagellar motor, the BFM. And what this motor can do is that it can rotate and generate torques. Okay. And when it generates torques, the flagella rotates as a consequence. The flagella can rotate either counterclockwise or clockwise. Okay. Now, if all of these bacteria rotate counterclockwise, they bunch up together and they bunch up together and they rotate counterclockwise. That is something like this. So you have one sort of thick helical filament that is rotating counterclockwise, or they could rotate individually clockwise, something like this over here. Okay. And depending on these two modes of rotation, you get two types of motion. Okay. So here is your bacteria, bacterial flagellar bundle. So the different flagella are sort of bundled up together and they are rotating in the same direction. As they rotate, they generate a propulsion and the bacteria moves. Okay. So excuse the quality of the video. So you'll see as the flagellum rotates, it generates a propulsion and the bacteria moves. Once in a while, you'll see that the flagella are unbundled and the bacteria is not really moving. When these flagella are bundled together, that is when the bacteria locomotes and it translates. And every so often, it will unbundle, and what it will do is that it will change direction. So when it is bundled up and it is rotating, it moves in one direction roughly. Then after some time, the flagella will unbundle, and then it will do what is called as a tumbling, in which it chooses a new run direction, and then it will go towards the direction of that new run. So here are trajectory traces of these E. coli bacteria. I think this is E. coli bacteria. So you plot basically the center of mass of this bacterial, single E. coli bacteria, and you generate trajectory traces like this. So what you'll see is that here is a sort of segment of this time series where the bacteria is running, so it moves in a line. Then after over here, this is where the flagella would have uncoupled and they would have been rotating individually. This is what is known as a tumble, and then it changes a new direction at random, and then again runs in that new direction. And then again over here, this run has ended, and it chooses a new direction and goes on and on. This is called as a run and tumble. This is called the run and tumble motion of bacteria. And this is sort of the prototype of when we talk about random walks in biology. This sort of run and tumble motion of E. coli bacteria is the sort of prototype motion that we have in mind.
Many species of bacteria quicker 100 times per second.
All right. So just to give you a sense, so these are the flagella in the E. coli, and if you zoom in into these flagella, you will see that. So these are the proteins that make up this flagella. This is called as the hook, and this is the filament. And inside the membrane is this rotary motor, which is actively driven by fluxes of ions across this membrane. And as it rotates, it generates a rotation of this flagella itself. It's very complex. It's a very complex protein assembly. It's made up of multiple proteins, each with its own function and so on. But essentially what it does is that it generates a torque, and this torque controls the rotation of this bacterial flagella.
And as its name implies, remains stationary. And the rotor, which rotates. The motor-derived flow through the mutational
so this is a lot of biology.
Although the molecular mechanism of rotation is not known.
But essentially, the bacteria can sort of control, the rotation of this flagella and thereby generate motion.
This binding causes a conformational change in mode A proteins, resulting in the first power stroke that moves the rotor incrementally.
Now, if you think about, so this motor therefore has two states, right? It can rotate clockwise or it can rotate counterclockwise, or the flagella rotation has these two states. It can rotate clockwise or counterclockwise, and it needs to switch from this one state to the other. If the switching is a random process with some rate r per unit time, right? What you would expect is that these interval distributions, that how long does it run before it goes into a tumble state, or how long does it tumble before it goes into a running state. This would be a Poisson process, right? Because that's what we learned. And indeed, if you plot these distributions of twiddle is what Berg initially called the tumbles. So nowadays we call it tumble, but anyway, it's the same thing. So tumble lengths or run length distributions, you'll see these are nice exponential distributions, as indeed you would expect for these interval distributions. Remember this P of tau we wrote as some r e to the power of minus r tau. So these are these interval distributions that you see in E. coli, which says that these at least in normal environments, and I'll say a little maybe about what normal means. In these normal environments, these switching processes are Poisson processes. You have some rate per unit time that you will switch from a counterclockwise rotation to a clockwise rotation, and that governs your run length distributions and your tumble time distributions. Furthermore, if you sort of think about, I said that so what a bacteria is doing is that essentially it's running. So when it is in the run mode, it goes in some direction. Then after some time, it stochastically switches to this tumble mode. When it's tumbling, it is not really going anywhere. It's just trying to sort of doing some very small sort of excursions, essentially staying where it is. Then it chooses a new direction to run, and then it goes, and then again it tumbles for some time, and then it goes, and so on and so forth. So you generate those trajectories that we saw. However, if you see these trajectories, these are not really straight lines, but these have some there is some drift of these while the bacteria is in the run phase. This is not really a straight line, but there is some drift, and that is because there is some rotational diffusion of the bacterial cell body itself perpendicular to the direction of motion. So if, for example, here is the bacteria is moving along the x-axis, it can rotate around, let us say, the y and the z-axis, and that will give rise to some rotational diffusion, and that rotational diffusion will give rise to trajectories such as this. In fact, this also Berg quantified, and he said that I think roughly over the course of a single run, you diffuse around 20 to 30 degrees, you diffuse an angle of around 20 to 30 degrees. All right, so this is in the course of a run. Now, there is of course changes of angles when you tumble. Okay, so you were in a run, then you tumble and you choose a new direction of a run. And you could ask that, what is this, if I looked at this change of angle, what is this tumble angle between two successive runs, so this one or this one or this one, what is the distribution of these angles? Now, if these stumbles were completely random, so you chose a direction at random, then you could ask that, well, what is this average tumbling angle, and what would that be if the tumbles were completely random? So I am moving along some direction, so let us say I am moving along the z direction, and I have to choose a new direction after tumbling, and I choose that direction completely randomly. What would be the average angle of tumble? Anyone? Huh? 0? Why? I mean, good. Yes? Huh? But you have to think that, how can you get different values of theta, right? So let me think about a unit sphere. Now, this value of theta means you could be in this concentric ring. A different value of theta means you could be in this concentric ring, and therefore the area that you are allowed to go to has a dependence on theta, right? Because these are 2 pi r sine theta d thetas, right? So what would be the average? Still 0? No, it would actually be pi by 2. So if you work out this area is 2 pi r, it's a unit sphere, so 2 pi sine theta d theta. The total area of the sphere is 4 pi r square, so 4 pi. So the probability that you uniformly, if you choose uniformly any angle, then this p theta is this by this. The probability of choosing an angle theta is 2 pi sine theta d theta by 4 pi, which is half sine theta. And then you can calculate this average angle and the variance of the average angles and so on. Maybe I'll work this out as a small assignment problem, if I remember to put it, and show that the average angle you would expect is 90 degrees, pi by 2. All right. So once you establish this, this is what you would expect, you can then go ahead and measure the angle distributions in these experiments. So again, Berg did that for these wild type E. coli bacteria, and what he found is if you plotted the angle distribution, so again there's a change in direction from run to run, so like this sort of angle distributions. It's not really 90, it's sort of biased slightly towards smaller angles, I think roughly around 60 degrees or so, plus minus something. So this flagellar change of angles is not completely random, so the flagella is actively doing something in order to determine, yes? Huh? So it is saying that what is the probability that I have tumbled by an angle theta. So therefore I lie in this region between theta and theta plus d theta. Okay. Now because this is also theta plus d theta, but also this and also so on. So how much of the area of this unit sphere can I access in order to have tumbled by theta? So that is that area. Always found in fluid.
Yes, sir.
Yes, yes, but why do you ask? So, I will clarify my answer a little bit. I mean, it can be a complex fluid in that it need not be standard Newtonian fluid. It can have, depending on the viscosity and so on, it can have different non-Newtonian responses. But yes, in principle, it's a fluid. So, but why in general? No, yeah. So, in that sense, it's always a fluid. The nature of the fluid depends because, like I said, the environments that these bacteria swim in can often be very complex. You can have other insertions and so on, which makes the stress strain response nonlinear, and that will govern the motion. But there is always a fluid, for example. Okay? And there is always a background fluid that this is swimming in. In fact, these fluids are generally what we call as low Reynolds number fluids. Where you can mostly neglect inertial forces and neglect inertial forces compared to viscous forces. Yeah, so it's always swimming in some sort of a background fluid. Right. So if you plot the probability distribution of these events, Berg showed that these are not exactly random, but the flagella is doing something actively in order to regulate this changes of direction from run to run. And again, this is not something that is set in stone. You can modulate it through various different physical or chemical cues, as I will show. Now, okay, so bacteria is swimming in these complex environments. Okay, and like I said, this swimming is this sort of a prototype of a random walk. This is the prototype of a random walk motion where it walks for some time, then chooses a new direction, and then walks in that direction, and so on. However, bacteria, of course, are living organisms, and it likes to find food. And by food, I mean essentially some chemicals that are necessary for its functioning. So, for example, this bacteria is swimming and you put a source of nutrients somewhere over here, let us say, okay, you put a source of nutrients over here, the bacteria would ideally, in order to survive, would like to sort of preferentially swim in this direction, okay, so that it can access this source of nutrients. Okay. And the way it does it is through these sensors on the surface of its cell, which are called chemoreceptors, and this sort of a motion taxis is motion. So, motion in the presence of a chemical is called chemotaxis. Okay. So, here is an example again. So, this is a different bacteria. It's not E. coli. It's a Helicobacter pylori. It's a bacteria that lives inside our stomach. It's a gut bacteria. And it can sense various different chemicals. So, in this particular case, the experiment is done using the chemical urea, and here is a wild type bacteria. Wild type meaning, so often you will see this term in biology, wild type, which means that it's a standard bacteria that you would find in the wild, and then you can do certain genetic mutations to produce mutant strains of that bacteria, and that is the two sort of panels over here. This is a wild type, this is a mutant. And this is one mutant in which you have knocked out a protein, TLPB. So, that is called as a delta TLPB, which means that you have knocked out this protein. And what they want to see is the response of the motion of the bacteria to this chemical urea in these two bacterial species. Okay. So, what you do is that you take a pipette and you start introducing urea through this pipette. So, this is my source of the chemical, and you will see that in the wild type what will happen is that bacteria try to swarm towards that source, right. So, whatever was the initial concentration of bacteria, you will have many bacteria trying to crowd towards that source, which shows that bacteria is sensing this chemical source and trying to move towards it. On the other hand, when you have this mutant where you have knocked out this TLPB, the bacteria continues swimming as it was before the introduction of the chemical. There is no aggregation of bacteria at this source point. And this TLPB is actually one of these chemoreceptors that are present on this bacterial cell surface and is responsible. So, therefore, what this sort of experiment shows is that this chemical, this receptor TLPB, this chemoreceptor is responsible for sensing this chemical urea and then modulating the motion of the bacteria in response to this chemical. You can do similar sort of experiments. So, therefore, the picture is that how does bacteria sense chemicals? You have a source. From this source, you get this chemical that diffuses out with some diffusion coefficient, let us say. These chemicals are sensed by receptors on the surface of this bacteria. And here what this experiment shows is that this particular protein TLPB is one such receptor, one such chemoreceptor. And if you knocked out this protein, it would not be able to sense this chemical gradient due to urea. Okay. All right. You could ask that is this the only receptor? And then people, after we put the chemical? Well, yes and no. So, in principle, you could have a biased random walk. So, I will talk about random walks in a bit, the mathematical structure, but let us imagine a random walk, right. So, this is what motion I was saying over here. So, I am a bacteria, at some point I decide that I will go in the front or to the left or to the right, and then I walk in that direction, and then I again choose. Okay. So, that is the pure unbiased random walk. Now, I could still make a choice, but I could make a choice with an enhanced probability in some particular direction. So, I am still tossing a coin, but now it is no longer a fair coin. Maybe 70% of the time I move forward, and the remaining 30% I move in the other 3 directions. So, it is still a random walk, but it is a biased random walk, for example, and then I could preferentially choose a direction. It could be other sort of walks, it could be other sort of walks as well, depending on what sort of whether the interval distribution remains exponential or not, and I will show you that it does not necessarily always remain exponential. Okay. But so, it is still a stochastic, it remains a stochastic process. The dynamics of this stochastic process can be different from what is the dynamics in a random walk. Okay, but yeah, go on. Okay, so it's like a chicken and egg problem. This is the sort of motion that the bacteria shows, and you can then analyze the motion and then try to understand the mechanics of the process that governs this sort of motion. Why has evolution optimized this motion? That's perhaps a larger question. I do not know, but it turns out that even in the presence of sources, there is a random component, and this randomness is often so this randomness or this noise is actually how do I put it? It's subtle in the sense that overall, if you see the motion, yes, it goes towards that, but if you were to look at each individual bacteria, you will see trajectories that are going away from the source as well. And there is a certain value to sampling the environment in order to survive, instead of just going in one direction. So, I do not have a clear answer, but evolution has come up with this strategy where it has some sort of randomness to the motion, but that randomness is modulated in the presence of sources. And then what one can do is sort of try to understand this sort of process as that happens. There is some value to this stochasticity. Maybe if at some point I will try to work out an example of why noise is beneficial in these sort of processes. So, what was I asking? So, I was asking that, so here are these receptors that senses these chemicals. Is there a single receptor that senses all chemicals? So, to answer that, you can do a different experiment. So, this is chemodetection of multiple signals, and here again this is pylori, H. pylori. Here in a pipette, what you will do is you will put a mixture of 2 chemicals, urea and hydrochloric acid, HCl. Urea is a chemoattractant in that it is known that these H. pylori bacteria want to move towards the sources of urea. On the other hand, HCl is a chemorepellent, which means it is known that these bacteria want to move away from this source of HCl. Okay. All right. So, now you have an experiment where you in a pipette you have a mixture of urea and HCl. So, an attractant and a repellent. Okay. You take these wild type bacteria and then you take this bacteria, mutant bacteria, where you have knocked out these chemoreceptors TLPB. What would you expect? So, forget about the mutant for the time being. In the wild type, what would you expect when you put a mixture of attractant and repellent? Remember, in the earlier case, when you just had an attractant, the bacteria, so here is my source and the bacteria all swarmed to the source. Okay, it just everything swarmed to the source. Here you have a attractant and a repellent, and what would you expect? Anyone wants to hazard a guess? It will move towards? It will move towards the attractant, but it's mixed together, right? So, when I put a pipette, the pipette has a mixture of urea and HCl. Huh? Correct. So, let us say, I don't know, let us say I take equal concentrations. Motion should not be affected. Okay, that's a valid guess, that motion should not be affected. Any other guesses? Their speed might decrease due to increase in number of what particles? You mean bacteria? Oh, you mean these the chemical signaling particles, the urea and HCl. So, their speed might decrease. So, you mean purely as because of something like excluded volume effect, like I have many ions that are now coming out of this source and that impedes the motion of the bacteria. Do you mean as a physical effect or as a chemical effect? As a physical effect. But generally, these ions are much smaller in these sort of experiments, at least. The ions are much smaller, the ions are much smaller than the size of the bacteria, and in these sort of experiments the cumulative excluded volume fraction would be too small to produce much of a physical signature due to self avoidance or anything. It would repel, okay. All of these are valid guesses. So, what it does is that it produces anybody else wanted to guess? What it does is that it produces a zone of exclusion. So, you will see the pipette, okay. So, now we introduce the pipette. What happens is that the bacteria want to move towards this, but then if it moves too close, then this chemorepellent comes into the play, and what it does is that it creates a sort of zone beyond which bacteria do not penetrate. So, if you are far enough, so if your bacteria is far enough, you sort of want to come close. If you get too close, then the repellent wants to push you back. So, effectively you get some sort of a zone of exclusion around this pipette source. The size of this zone of exclusion you can play by changing the concentration, the ratios of the attractant to the repellent. If you put more attractant as compared to repellent, this radius will get smaller. If you put more repellent as compared to attractant, this zone of exclusion will get larger. Okay. They are of course different because these are different chemicals, so they will have different diffusion coefficients. Roughly, although having said that, I do not know the exact numbers, but order of magnitude I would expect them to be similar, a few microns squared per second, that's roughly. But yeah, I do not know the exact numbers, but of course precisely they would be different because these are different chemicals. Both the chemicals will? So the pipetting experiment, of course, if you just put a spot and let it be, it would disperse, but the pipetting experiment is that you keep on pipetting this chemical at a certain rate, so that there is a continuous source wherever your pipette is, at least for the duration of this experiment. No, so that's what. So you are introducing a chemical at some finite rate, okay. These chemicals are going to diffuse, okay. And they are going to get absorbed, of course, because you have these bacteria which are taking up these chemicals. So eventually you will get some sort of a steady profile. some sort of a gradient of these concentration profiles of these chemicals. Okay. And in response to that, what you will get is, after you have waited for some time, you will get sort of a roughly constant zone of exclusion for a given chemical composition of this urea plus HCl. So, you will get some sort of a steady state essentially. Yes, it will. So, it does not matter whether it is ultimately it is a chemical. Okay. It comes to the chemoreceptor, the bacteria takes it in and then modulates its motion in response to this chemical event. Because unless it sees this repellent and it sees it essentially by taking it through this receptor and then modulating its response, it does not know whether to go towards it or away from it. So, of course, there is uptake of both attractant and repellent. How it modulates its behavior is different for these two different classes of chemicals, right. Now, I do not know if you focused on this mutant. So, what happens in the mutant? Let us see. So, you will see what happens in this delta TLPB mutant is that the repellent seems to win over, right. All the bacteria are cleared from the field of view, which means that the attractant is not playing any role, but the repellent is sort of pushing away all of these bacteria. So, having done this experiment, what would you say about these receptors? So, would you say that this TLPB receptor is responsible for both detecting urea as well as HCl? Yes or no? Yes? Okay. Why? So, I have knocked out the TLPB receptor. Okay. The bacteria still continues to sense the repellent, but it does not seem to sense the attractant anymore, right. So, then what is the conclusion? So, therefore, what they deduced was there are different kinds of receptors and different receptors sense different molecules. So, this TLPB receptor is the one that is sensing urea, the attractant. Maybe there is a different receptor, whatever it is. It turns out to be something called a combination of A plus D, two types of receptors in concert, which detects this HCl. So, if you knock out these TLPB receptors, the bacteria cannot sense the urea, but it can still sense the other one, and therefore, it still pushes away, it still goes away from the source. So, there is a way for bacteria to detect multiple signals at once and then modulate its motion accordingly to these multiple signals. If the bacteria come very close to, okay, correct. So, at least initially you will get some bacteria which are very close to the source. After some time, of course, most of the bacteria have moved away, but initially when you put the pipette, you will have bacteria which are very close to the source, and you are asking that what happens to the bacteria if it is ekdam at the point of the source. I do not know what HCl does. I do not think it directly kills the bacteria, but it may kill the bacteria. So, I will have to check what the chemical exactly does to this pylori. But a single molecule absorption is not going to kill it, that much is so it is not that you put one, it is not toxic in the sense that you put one molecule on this receptor and the bacteria dies, because it is always sensing these molecules. But if you trap it inside a zone of high HCl, which is I think essentially what you are asking, I will have to check what the response of the bacteria is to prolonged exposure to HCl. Yes. So, that is a interesting question. Maybe we will talk about it at some point. That is there a threshold beyond which it can detect signals, right? So, that has to do with the, I am trying to remember the answer actually. So, there are of course multiple receptors on the surface of a cell. Okay, there are multiple of these protein receptors that sense one type of chemical. Collective effect or collective is sensing between all of these receptors sets a threshold, but I am trying to remember if an individual receptor, how sensitive is an individual receptor, and whether it change like a single molecule detection change. I think so, but I'll check it. I'm not sure. I think so, but I'll check. Oh, from the source. Yes, of course, but the question is that, of course, my source is here, but I think the way at least I am interpreting this question is that my source is somewhere, but one molecule has diffused to this receptor, and then is it sensitive to detect that one molecule or not, or does it need to have 10 detection events? Like, we are talking in the case of photons, that how many photons do you need to in order to detect? One could interpret this question in a similar light. I'll check and see. So this is in a sense the cell signaling problem. Cell signaling problem in that you have these receptors. So this is this particular receptor TLPB, this structure that this receptor that I was talking about. This is the protein structure of this receptor, and here is an urea molecule which binds in this pocket. There are many such receptors spread out on the surface of this cell. Okay, and then related. So one sort of a question is the motion of the E. coli in the presence of these receptors, in the presence of these chemicals. The other is, of course, the motion of the chemical molecules itself to reach the cell body, like I was saying. So you have this source over here, and these chemicals have to diffuse, and then they have to reach this cell body. So you can invert the problem that if I fix my frame of reference to be on the E. coli, and I look at molecules coming from a distant source to these receptors on the surface, then what is the statistics of these arrival events? Okay, so there is another sort of problem, which is the cell signaling problem, and again that is something that we'll look at in maybe the next couple of lectures. So this is, I've already talked about this, whether it has one receptor or many receptors. This sort of stills of this image, which shows that you can characterize this, like I said, this depletion zone around the source by playing with this concentration of HCl and urea. This is the TLPB mutant where it does not feel the attractant, but it feels the repellent, which is why you cleared the field of bacteria. Here's a different mutant, which is a completely, you stop chemotaxis, you stop the motion, and there neither the attractant, so in this mutant bacteria, so it looks just like once you input this chemical signal, the bacteria continue to do what it did without detecting the source at all, because this mutant does not detect either the attractant or the repellent. So this is a different sort of mutant. So you can get a whole gamut of behavior depending on how you tinker with these bacteria. So in that sense, these E. coli, so like I said, these E. coli are very nice systems because you can produce these genetic mutants in order to sort of tease out the effect. So we have a physical process which is chemotaxis, so movement of bacteria in the presence of a chemical signal. You can tease out the sort of bacterial, you can tease out the biological ingredients behind this process by doing these sort of controlled experiments where you knock off one protein and see what that does, you knock off a different protein and see what that does, and thereby you come up with a hypothesis of how these bacteria move. Like I said, so these swimming strategies are not set in stone. Depending on different chemical or physical feedback, you can modulate these swimming strategies. So for example, let me just maybe show. So this is an E. coli swimming in some sort of a micro channel. This is an E. coli swimming in some sort of a micro channel, which is of 50 micron width. Okay, and then you put it in a very narrow channel, which is of 10 micron width, and you put some sort of chemical source. You can change these run length distributions. So like I said, in a Poisson process, the run length distribution is exponential. So that is this over here. If you see, this is a semi-log plot to this sort of a scenario where you have a chemical gradient and you have a physical confinement. You'll see that the run length distribution is switched to a power law. So this is a log-log plot, and it's a straight line in a log-log plot, which means it's a power law distribution. And this sort of walks are, so these sort of walks are called Levy walks. And you can see that sort of that signature of that even in these tumbling angle distributions. So like I said, so think of this as sort of bulk motion. This is 50 micron without any chemical gradient. You have large angle distributions around the 60 degrees or so. On the other hand, if you put this under confinement and you put it under a chemical gradient, you'll suppress. So you compare this plot of these, this plot of angles with this plot of angles, you have suppressed a lot of these large angle tumbles, so that the bacteria is moving mostly in the forward direction. So you can play around with these, swimming strategies. How often does it switch from run to tumble? How does it sample these angles that are available to it by modulating various physical and chemical factors, such as confinement and such as strength of the chemical gradient and so on. All right. So having said that, so having given you a sort of background of why this sort of motion is important in a biological context, let's get to a little bit of random walk and we will see how much we get to today. Okay. So random walks. Okay. So the prototype that I have in mind is this sort of an E. coli, so I walk, I choose a new direction, I walk, I choose a new direction, I walk. So the simplest possible random walk that we can define is, let us say, in one dimension. So let us say in one dimension, one dimensional random walk, and in order to simplify this even more, we can choose a discrete time and a discrete space random walk. So a discrete time plus a discrete space. Okay. What does it mean? It means that because it is one dimension, I have motion on a line, right. Here I have my random walker. Okay. Discrete space means that it can, there are sites on this 1D line that it can hop to, right. And discrete time means that every time it hops, you increment your time with some discrete time interval. So your random walker was here, let us say, it can hop to the right or it can hop to the left, and I put probabilities for this. Let me say I put a probability to the right, that let us say I have a random walker here, it can hop to the right with some probability p, and it can hop to the left with some probability q is 1 minus p. Okay. And what I want to ask, and every time it hops, I increment my time step by 1, so delta t equal to 1, let us say. And then I want to ask that, what is the probability distribution, what is the probability that my random walker is at some position r after n steps. Okay. So that is the essential quantity that I want to determine. So I have this one dimensional random walk, discrete space, discrete time. This is my time index n, because there is n steps. This is my space index r. Okay. And I can write a sort of recurrence relation of this Pn of r by asking that where could it have come at the nth step. If my random walker is at r, then it could have come from 2 positions at the n minus 1th step, right. So it could have come from 2 positions at the n minus 1th step, which are r minus 1 and r plus 1. So I've said that these delta x's are also 1. So whenever you hop one time, one step, 1 unit of space. Okay. So at the nth times, at the nth step it's at r. So therefore at the n minus 1th step, it could have been at r minus 1 or it could have been at r plus 1. Is this correct? Yes. Thank you. It's not correct. Who said it's correct? So where will p be? First term, because this is a hop to the right from r minus 1, it's coming to r. So that's p, and then this is q, right. If you have p is equal to q, then this is an unbiased walk. It is an unbiased random walk. If p is not equal to q, then this is of course a biased random walk. You will have a net sort of drift in one direction, right. All right. And what I want to do is to sort of solve, I want to ask in general, what is this Pn of r, given that it satisfies a recurrence relation like this. So, remember we studied about when we were talking about probability distributions a couple of lectures back, we defined characteristic functions, right? We defined characteristic functions which were what? Which were the Fourier transform of the probability distributions. And what did the characteristic function do? The characteristic function generated for us moments of the distribution, and knowing the probability distribution is equivalent to knowing all of its moments. So, we can try to calculate the characteristic function. So, I will call this g n of k, it is e to the power of I k r p n of r, right, integral dr, correct, from minus infinity to infinity. So, here is my Fourier transform, the characteristic function, e to the power of I k r p n of r, and again what we had done was to sort of expand this exponential in the series form, and therefore show that when you looked at powers of I k to the power of n, those corresponded to the nth moments r to the power of n p n of r. So, that is how we generated the moments, and therefore, if we know the characteristic function, we can know the moments, and in general it is the derivative. So, if I want the mth moment in this case d dk, I look at the mth derivative of g n of k, and that gives me average r to the power of n, right. This is evaluated at k equals 0. Okay. So, the characteristic function generates for us the moments, and knowing the moments allows us to reconstruct the probability distribution. All right. So, I want to find the characteristic function of this p n of r, let us say. Okay. So, I write g n of k is equal to integral dr minus infinity to infinity e to the power of I k r p n of r, and in place of p n of r I substitute this relation. So, I have p n minus 1 of r minus 1 plus q p n minus 1 of r plus 1. Huh? Does not matter. Whichever, actually you will see all sorts of is minus, plus. When you do the reverse, you just have to keep the correct sign correctly, so that you get the p of r. So, if I use plus, when I do the inverse, I will put a minus. If I put a minus, when I do the inverse, I will put a plus. But anyway, okay. Yeah, sometimes because I have made my notes at different times, I will have switched between conventions, but it does not matter. All right. So, yes. You could, in principle. I will give that as an assignment, and then you can see what happens. So, what Jeremy is asking is that in principle, I could choose it hops to the right with p, it hops to the left with q, and it stays at the same place with r, which is 1 minus p minus q. Okay. You could do that. It's worth doing. It's worth doing. So, I'll give it as an assignment. It won't make any difference. That's the short answer. But it's worth doing and satisfying yourself that it won't make any difference in the continuum limit. Okay. So, here is my Pn of r. So, I substitute for that over here, and then let me try to massage this a little into a little more usable. So, let me say I do a p e to the power of minus I k, no, p e to the power of plus I k, then I do e to the power of I k r minus 1 p n minus 1 of r minus 1. Okay. So, I divide and multiply by e to the power of I k. So, this becomes an I k r minus 1. So, that is one term, and then I do the same thing for over here. I do a q e to the power of minus I k, then I do e to the power of I k r plus 1 p P n minus 1 of r plus 1. Okay. Let me just separate out the integral. So, I do a dr over here minus infinity to infinity. Okay. So, this term is of course does not depend on r. So, I pull out the p e to the power of I k, and then I am left with this integral dr e to the power of I k r minus 1 p n minus 1 of r minus 1 minus infinity to infinity for the first term. What is this? This is g n minus 1. So, this is p e to the power of I k g n minus 1 of k. What is the second term? What is the second term? So, q e to the power of minus I k into again g n minus 1, right. So, g n minus 1 of k, right. Because whether you use the substitution r goes to r minus 1 or r goes to r plus 1, the dr does not change, the limits still stay minus infinity to infinity. So, both of these integrals are g n minus 1s. So, essentially what you have is for g n of k is this, let me write here, is p e to the power of I k plus q e to the power of minus I k of g n minus 1 of k. Okay. Is this fine? Okay. Let me call this u of k, this factor, u of k g n minus 1 of k. So, what do we have? We again have a recurrence relation that g n of k is u of k times g n minus 1 of k with u of k given by this. So, I were to do this operation once more. So, now for g n minus 1, I write down the recurrence relation for p n minus 1 in terms of p n minus 2's at r minus 1 and r plus 1. So, what would I get if I did this again? I would get u of k whole square g n minus 2 of k, right. It is essentially the same procedure that I am doing. And if I did this again and again, what I would get is this whole thing. If I did this operation n times, I would get u of k to the power of n g 0 of k at time step 0. What is this characteristic function at time 0? Is this fine? Alright, so I have this. So, g n of k is u k to the power of n g 0 of k with u of k given by p e to the power of I k plus q e to the power of minus I k. Okay. Now, I need my initial condition, right, and let us say I choose my initial condition that the particle starts at the origin. Okay. So, I have this line where particle starts at x equal to 0. What does that mean? That means that P0, the probability distribution at time 0 for location r is a delta function, because this discrete space I use a Kronecker delta. So, delta r comma 0. So, it is 1 if r is at the origin, it is 0 otherwise, right. So, this tells me that the particle starts at origin. So, the particle starts at origin. That is my P0 of r, which means I can now calculate my generating function g0 of k, which is integral dr e to the power of I k r. Did I use minus or plus? I think I used plus. So, e to the power of I k r Pn of r, which is delta r comma 0, which means this is minus infinity to infinity. So, this is what? This is just e to the power of 0, which is 1, right. So, this is my initial condition that g naught of k is, if your particle starts at the origin, that initial condition means that your G naught of k is 1. So, then you put G naught of k equal to 1 and you have solved for your Gn of k, right. So, your Gn of k is just uk to the power of n, where your uk is this. Once you solve for your Gn of k, it means that you can now do the inverse Fourier transform and obtain what is your Pn of r, right. Okay, so I want my Pn of r, right, which is 1 over 2 pi integral dk minus infinity to infinity. I use e to the power of plus then, so I use e to the power of minus here, e to the power of minus ikr, Gn of k, which is nothing but u of k to the power of n. So this is p e to the power of ik plus q e to the power of minus ik whole to the power of n, right. So this is not minus infinity to infinity. And I can now expand this. So I write 1 over 2 pi minus pi to pi dk e to the power of minus ikr sum over s equal to 0 to n ncs p e to the power of ik to the power of s q e to the power of minus ik to the power of n minus s, right. And yeah, that's it. All right, so this is fine. I just expand that in the binomial series, and then I flip this summation and the so where do I have my k's? This is the inverse Fourier transform, right. So the my k is my wave number, wave vector, right. Isn't this the standard inverse Fourier transform? Huh? You stay as a? Let me just do this and then I can come back to this. I just want to finish this before this ends. So what I want to do is that I want to separate out my terms which don't depend on k. So I want to separate out my s equal to 0 to n ncs p to the power of s q to the power of n minus s, and then I do this integral dk by 2 pi e to the power of minus ikr e to the power plus iks minus ikn plus, so 2 iks essentially. Okay. So this is, if I just simplify this, this is e to the power of minus ik n plus r minus 2s, and this is essentially your, this is essentially what? This integral. This is again your delta function. This is a delta of n plus r equal to 2s, okay, which means that what I can do is substitute, so s equal to 0 to n ncs p to the power of s, so s is n plus r by 2, n plus r by 2, and q is n minus r by 2. Q is n minus r by 2, right? Is that fine? No, it's not fine because I don't have the summation anymore. I had the summation, I write the delta function over here, then I do the sum. So when I replace this s, that summation goes away. So, this is just NCS, right. So, I recover my binomial distribution for this Pn of R. So, I recover my binomial distribution for my Pn of R. This is NC n plus R. So, we started from this one dimensional hopping process essentially on discrete space and time. A particle could hop to the right or to the left. We specified an initial condition that the particle starts at the origin at time t equal to 0, and what we can do is that we can solve exactly for the probability distribution of the particle to be at position r after the nth time step, which is given by this binomial distribution. Now, you can do one more thing, which is that you can take the limit of the large n limit. So, you can take the limit that n goes to infinity. You can take the limit that n goes to infinity, use Stirling's approximation for all of these factorial terms that will come over here, and then show what. So, if I take this limit, what will this Pn of R go to? Anyone? A Poisson distribution? Poisson distribution, anybody else? So, I have the binomial distribution, I obtain this exactly. Now, I want to take the limit that n goes to infinity and ask what is this, what does this Pn of R become in the limit that n goes to infinity. Any? A distribution. What do you mean the distribution will approach 0? P and Q are some hopping rates. I mean, it's some probability to hop to the left or to the right. That is given, that is the problem statement in that sense. Anurag? Take the binomial, take a probability distribution like this, which consists of steps at every time you take an independent decision, which is you hop to the right or to the left with whatever probabilities P and Q. You do that many, many times. Each decision is independent of the other. In the limit n goes to infinity, it's a Gaussian distribution, right? Why? I thought Amitabh was teaching this, no? This is the central limit theorem, right? So, you will go to what? You will go to e to the power of minus r minus n times p minus q, right. If you have a biased random walk, your mean stays at 0. If you have a, if you are an unbiased random walk, your mean stays at 0. So, if p equal to q, it's just and then whole square divided by 2n, that is your variance. And so, you should try this in case you have not. That if you take the Gaussian, if you take the binomial distribution and then you take the large n limit, because the central limit theorem implies that this binomial will converge to a Gaussian with mean n times p minus q and variance n. Yes, over here. Here, yes. The power of q is also n plus. So, I have n minus s and my delta function is n plus r equal to 2s. So, n minus n plus r by 2. All right. If I understand, at least Amitabh has been doing, starting from the other approach, which is writing down the master equation and then looking at specific cases, of which the discrete random walk may have been one of the cases. This is a sort of other approach where you start with the discrete time, discrete space random walk, and you can work out that exactly, at least in one dimensions. But now you can take the continuum limit of this. You can ask various other questions as well. For example, you could ask that, I have started my random walk from the origin over here at 0. If I waited long enough, will I come back to the origin or not? Okay. So this is a property called recurrence of random walks. And again, that is something that you can work out exactly for these sort of walks. So that's what we'll do next. But you can also take the continuum limit of these, so the continuous space, continuous time limit of these discrete processes, and thereby go to the diffusion equation, okay, which governs, and then you can use the diffusion equation to analyze various bacterial systems such as E. coli motion, such as other examples that we'll talk about as well. Okay. This is sort of the other approach that one can take. So I'll stop here for today. I'll talk about this recurrence and transience next day, and then take the continuum limit of this random walk.